Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the transgenic edible vaccines. So, friend, this is also a very important topic for all the competitive exams. And in this lecture, we will discuss about the several facts of edible plant vaccines. So, let's start today lecture with the edible vaccine. That what are the edible vaccines? So, edible vaccines are genetically manipulated food. This is also known as GM food, genetically manipulated food or genetically modified food containing the organelle, and these have foreign organelle or their antigens or related antigens that may provide active immunity against infections. It could be some viral infections or diseases or these. So the edible vaccine is known as genetically modified food which contain foreign antigens and provides active immunity against infections. So immunity, how they provide immunity? They activate the your immune system. And you know that antigen antibody reactions, that when you insert the antigens of foreign organelle, it bind with the antibodies of your cells and activate some reactions which which activates your immune system and provide immunity against the infections. So these are also known as transgenic edible vaccines. So plants are used as a bioreactors. What is the means of bioreactor? Bioreactor means as a reactor or system which can produce in the large quantity. So plants are used as a bioreactor to produce large quantity or to express antigen proteins of foreign organelle induced by the plant transgenic vectors. So in the last lecture of transgenic, we discussed that there are so many vectors like plasmids, bacteriophage. These vectors are used for the carrying the foreign organelle or your antigens and to produce certain special vaccines with high anti-disease ability. So आपका एडिबल वैक्सीन क्या होता है? ये एक जेनेटिकली मॉडिफाइड फूड है, इन्हें ट्रांसजेनिक एडिबल फूड्स भी बोलते हैं। इनमें क्या होता है, एक फॉरेन से हम एंटीजन डालते हैं जो आपकी बॉडी में आके आपकी एंटीबॉडीज को एक्टिवेट कर देता है और जो इम्यून सिस्टम है उसको बूस्ट कर देता है, विच प्रोवाइड द इम्यूनिटी अगेंस्ट द योर वायरल डिजीजेस। एंड द प्लांट्स आर यूज एज बायोरिएक्टर्स, मींस लार्ज क्वांटिटी में प्रोड्यूस करने के लिए प्लांट्स यूज करते हैं। एंड वेक्टर, वी आर यूजिंग वेक्टर्स सच एज प्लाज्मिड्स। इन वेक्टर्स में आपका जो ये है, क्या कहते हैं, जो एंटीजेंस जीन है उसको हम इसमें कैरिंग करते हैं और ट्रांसफॉर्मेशन मेथड से उसको ट्रांसफर प्लांट्स में कर देते हैं। एंड व्हाट आर द ग्लोबल नीड्स? सो यूनिसेफ एस्टिमेटेड दैट approximately 34 million children are not reached by the routine immunization, and it is also accepted that the expense of stabilizing conventional manufacturing units has hindered the development of market for biopharmaceuticals and biotechnology derived biologics. तो इसका मतलब है every year almost 34 million children are not getting their routine vaccines or immunizations. Also, it is accepted that the conventional manufacturing methods are not sufficient to fulfill the global needs. That's why therefore need of new pharmaceuticals and biotechnologies vaccine which can fulfill our global need. So this is the our need of developing this edible vaccines. Now come to the process of producing transgenic vaccines. So what are the process? So the first process is the isolation of antigen of interest. So in the first process, we isolate the antigen of interest. So in this process, mainly genetic engineering is involved. Genetic engineering. So in which we use some restriction endonuclease and what is the function of restriction endonuclease? This enzyme cuts the DNA into fragments, and second enzyme is the ligase enzymes. This enzyme add these fragments. So by the using of this genetic engineering, there will be an isolation of this antigen of interest and the structural gene or DNA fragments which encode in the epitopes. And what is the epitopes? This epitopes is the part of antigen. Epitope is the part of antigen which activates the immune system or antibody, because antibody have receptor for this your epitopes. Okay, then there are two methods that transform. These are the two transformation methods, and these structural genes transfer into the transformation vectors. Already discussed that your plasmids and bacteriophage is work as a transformation vectors, and this transferred by the two methods. First is the direct method or gene gain. This is also known as biolistic method, means by using the gene guns your edible your antigen is transferred between the plants. Second is the indirect method, which includes the agrobacterium mediated and most commonly used method, and it causes the stable integration of genes into the plant genomes, and here directly transient expression of your foreign antigen, and final step is the extraction of foreign antigen. So this is the process of producing transgenic. मीन्स सबसे पहले आपने एंटीजेंस लिया, उसको आपने सबसे पहले क्या किया, रेस्ट्रिक्शन एंडोन्यूक्लियस से कट किया, लाइगेज एंजाइम से उसको बाइंड अप कर दिया। बाइंड अप करने के बाद जो ओनली एपिटॉप पोर्शन है उसको आपने प्लाज्मिड में ट्रांसफर कर दिया। करने के बाद आपने डायरेक्ट या इनडायरेक्ट मेथड से उसको इंटीग्रेट कर दिया प्लांट जीनोम में, फिर उसके बाद उसका मैन्युफैक्चरिंग या उसका एम्प्लीफिकेशन स्टार्ट किया। सो दिस इज द प्रोसेस ऑफ प्रोड्यूसिंग ट्रांसजेनिक वैक्सीन्स, एंड दिस इज आल्सो वन ऑफ द न्यू मेथड to producing transgenic edible vaccines, which is via the plant virus. So you know that virus is having capacity to high replication, means they have very fast replication capacity. So first process is the development of recombinant virus, means in the virus we include the genome or antigen of interest. So in the process, we insert the antigen of interest. So this is called as recombinant virus, like recombinant plants. Then there will be a viral infection in the plant. This is the second process. In this process, virus replication take place, and this virus infection cause the expression of your antigen of interest protein or peptides, and by this vaccine epitopes can be synthesized and accumulating by changing the viral capsid proteins. So like that we are developing this recombinant virus. So this is the process of developing transgenic plant virus. So first process, we are developing the recombinant virus by the changing capsid proteins in the your virus. then there will be a infection in the plants. This cause the viral infection, and virus replication multiplication take place, and final process is the express of this your antigen of interest. So this is the process, and several viruses like CMV, TMV, tobacco mosaic virus, like that Gemini virus is used for the development of edible vaccine. So this is, there are three process. First by the direct transfer, indirect transfer, and this transgenic plant viruses. Now the production of vaccine have, there will be a four process. So first process is the isolation, in which the genetic engineering involved. Already discussed in the last slides that how genetic engineer isolates the antigen of interest. And second process is the propagation. So in this plant system used for the propagation and multiplication. And third process, in this we purify the edible vaccine by removing the secondary metabolites. You know that plant have several secondary metabolites which have impacts on the quality of vaccines. So they will be removed before the purification process. And fourth one is the formulation of your edible vaccine. So this is a common process of development of or production of vaccine. तो पहले प्रोसेस में आप आइसोलेशन करोगे, दूसरे में उसको प्रोपोगेशन करोगे, फिर आपको क्वालिटी को इंप्रूव करना बाय रिमूविंग द सेकेंडरी मेटाबोलाइट्स, और फोर्थ में आपको एक जो प्योर वैक्सीन है उसका फॉर्मुलेशन आपको प्रिपेयर करना है. दिस इज द फोर्थ प्रोसेस. एंड व्हाट आर द लिमिटेशंस ऑफ कन्वेंशनल वैक्सीनेशन, दैट व्हाई वी नीड transgenic vaccines? Because the earlier system, that is conventional vaccination, have many limitations. Like that, first is the safety concerns. You know that earlier developed that conventional vaccination is less safer than the modern transgenic vaccine, because there will be a failure in the inactivations which can cause the reversion of virulent form. मीन्स अगर हम वायरस जो आपका वायरस से यूज कर रहे हैं, अगर वायरस का मे बी पॉसिबल कि अगर इनएक्टिव फुली इनएक्टिव नहीं हुआ जो आपका विरुलेंट जीन है, तो मे बी पॉसिबल दैट इन सम केसेस दे विल बी अ रिवर्सन ऑफ द विरुलेंट फॉर्म ऑफ वायरस contaminated due to the less quality control, and there will be a possibility of contamination. And third point, there will be a need of refrigerator for the storage. And fourth one is the injectable vaccines only stimulate systematic humoral response. And fifth point is the sometimes so local inflammation, means they will cause some allergy, and some cause there will be a huge allergy. It affects your health, and not possible to develop vaccine for all disease by conventional method. And conventional method we cannot develop the vaccines for the older disease, and the process is also very slow. So there will be a need of generation of or producing of transgenic edible vaccines. So safety concern, contamination, need refrigerators, only some systematic response, and due to local inflammation and time taking, there will be a need of new method that is known as transgenic edible vaccines. Now we are going to discuss about the transgenic plant vaccine expression system. So selection of plant material which can both express high level of chosen vaccine candidate or antigen of interest, or be suitable for the extensive storage, means your durability, and the easy to take or oral delivery. So these are the some specific criteria for a ideal vaccine. तो क्या क्या होना चाहिए? एक तो वो हाई लेवल में एक्सप्रेशन होना चाहिए, मीन्स हाई क्वांटिटी मिले. सेकंड, वो एक्सटेंसिव स्टोरेज, मतलब वो ड्यूरेबल हो. एंड थर्ड, कि वो इजी से आपका मतलब ट्रांसफर भी हो सके, डिलीवरी भी हो सके, एवरीथिंग विल बी इजी. So these are the three characteristics of your ideal plant vaccines. So first system which used for the development of transgenic plant vaccine, that is your vegetables. So vegetables are used as a receptor of plant derived vaccines because many of them are appetizing, फ्री ऑफ टॉक्सिकेन मीन्स इनमें आपका टॉक्सिकेन नहीं होता है, फुल ऑफ न्यूट्रिएंट्स मीन्स आपका जो वेजिटेबल्स होता है वो फुल ऑफ न्यूट्रिएंट्स होता है, फ्रेश एडिबल्स मीन्स जनरली जो फ्रेश वेजिटेबल्स है दैट आर यूज फॉर द ईटिंग पर्पज. तो दैट्स व्हाई दिस वेजिटेबल्स आर ए good system for the expression of these plant vaccines and developed for the tetanus, diphtheria, hepatitis B and norwalk virus. So for this virus there are vaccines this developed, and potato is considered as an optimal model system on research on plant oral vaccines, and tomato is new expression system developed. So potato is the first vegetable which used for the development of plant vaccines, but nowadays tomato is also used as a new expression system for the development of this your plant vaccines. And antigen encoding hepatitis, HIV, pneumonia, septicemia, bubonic plague, rabies have been successfully transferred to the tomato. Means for this, by changing the crop capsid proteins, antigens for these diseases are transferred in the tomato. Next, the latest discovery of seed-specific promoters converts a target protein in seed, so high expression proteins are available because the seed, a plant produces the number of seeds. So seed number is very high, so high expression proteins can be developed through the seed proteins, and high level of recombinant protein expression was observed in proplastid of culture cells, cultures carrot cells. Means the third vegetables which used for the development of edible vaccine is the carrot, and in the carrot the protoplastid culture is used for the production of edible vaccines. And oral delivery of therapeutic protein by edible carrot preserved in proplastid of culture carrot cells, and the structural integrity of their target protein are as no cooking needed. तो जो आपका वेजिटेबल सिस्टम लिया गया था उसमें सबसे मेन प्रॉब्लम था कि जो हम वेजिटेबल्स यूज करते हैं वो मेनली आफ्टर कुकिंग यूज करते हैं, एंड वी कुक द योर वेजिटेबल्स, देयर विल बी चांसेस दैट देयर विल बी प्रोटीन डीनेचुरेशन. मीन्स जो आपका प्रोटीन है उसका स्ट्रक्चरल लॉस हो जाएगा. तो जो आपका रिएक्टिविटी है, जो एंटीजन फॉरेन एंटीजन है या प्रोटीन्स है, उसका एक्टिविटी आपका लॉस होने के चांसेस है. दैट्स व्हाई द कैरेट इज ए गुड वेजिटेबल और आइडियल वेजिटेबल सिस्टम फॉर डेवलपिंग योर एडिबल वैक्सीन्स. तो समझ में आया कि आपका जो पोटैटो है, टमाटो है, कैरेट सेल्स है, वो आपका डेवलपमेंट ऑफ एडिबल वैक्सीन्स के लिए यूज किया गया था, बट प्रॉब्लम इज द ओनली कुकिंग. That's why the carrot is a good source of developing edible vaccine from the vegetables. And next point is the fruits. And banana is one of the earliest fruit used for the transgenic programs for development of edible vaccines. Embryonic callus of papaya was genetically transferred with the different construction including the reported genes, GUS genes or biallelic means direct method. तो इसमें आपका डायरेक्ट मेथड यूज किया गया था डेवलप के लिए फॉर हेपेटाइटिस एंटीजेंस. हाउएवर फ्रूट्स आर वेरी पर्शिएबल मींस द कीपिंग क्वालिटी ऑफ fruits are less. That's why this system is also not good for production of high development of high edible vaccines. Means keeping quality is less in fruit, and also they are only seasonal fruits. Means fruit and vegetables are only seasonals. Means in whole the year we cannot get the antigens or your edible vaccine from these sources. Now the third system which are used for the development of edible vaccines, which is crops. So our agriculture is crops is used for the development of edible vaccines. So spermatocytes are more suitable for oral delivery vaccines because they have abundant soluble proteins. तो आपका जो सीड्स है, क्रॉप्स है, उनमें आपका जो सोल्यूबल प्रोटीन है दैट इज वेरी हाई, एंड आल्सो दे हैव वेरी हाई कीपिंग क्वालिटी. They have very high keeping quality. But still there is a problem. First, we are not cooking the food directly, means we need some cooking process. And second, they also seasonal and they also take time to produce. So these are also problem for these crops. And alfalfa is perennial crop with strong regenerative capacity and propagation, which continuously produce large clonal population by stem cutting. तो जो आपका अल्फा है उसमें क्या है कि आपका जो मीन्स इसमें सीजनल का प्रॉब्लम नहीं है, मीन्स दे हैव स्ट्रॉन्ग रीजनरेटिव कैपेसिटी, मीन्स दे आर perennial in nature, and also they are reproduced by stem cutting. So you can produce the many clonal cells. So this is also a good system for expression of edible vaccines. And glycoprotein E2 expressed in the lettuce for the classical swine free hope pest virus was developed. तो आपका जो लेट्यूस है उसमें एंटीजन ट्रांसफर किया गया था ग्लाइकोप्रोटीन E2 for the virus your swine fever hope. And the low level of secondary metabolites and high protein content in alfalfa leaves make it a good bioreactor for the production of recombinant protein. तो जो आपका अल्फा है that is good source for the development of edible vaccine because it have less secondary metabolites, high protein contents, and for this we can use for development of edible vaccines. And the new system that is made commercial platform for the प्रोडक्शन ऑफ फार्मास्यूटिकल्स एंड रिकॉम्बिनेंट एंटीबॉडी. तो अभी रिसेंटली मेज भी आपका यूज किया गया है फॉर डेवलपमेंट ऑफ एडिबल वैक्सीन्स. एंड सम प्रोटीन्स हैव बीन एक्सप्रेस्ड इन द राइस आल्सो व्हिच हैड वेरी हाई लेवल बाय यूजिंग कॉन्स्टिट्यूटिव एंड एंडोस्पर्म्स प्रोमोटर्स. मींस आपका एंडोस्पर्म में आपका जो जीन एंटीजन ऑफ इंटरेस्ट है वो आपका कहाँ ट्रांसफर किया गया है, एंडोस्पर्म्स में. And cereal crops are also ideal because the endosperm are full of soluble proteins. Means जो आपका endosperms है सीड का उसमें आपका soluble proteins बहुत हाई होता है and can be easily separated and enhance the antigen concentration and reduce the oral dose. Means आपको एज ए वैक्सीन की तरह नहीं लेना है, जस्ट ईट करना है. So these are the some advantage of edible vaccine from the crops. Also there are some new systems like algae based edible systems. So in this some algae like that Chlamydomonas, Amphibium, Symbodinium, Slendrothecia and Pheodictyum, these are algae used as a model system for the edible vaccines, and it solves the problem of low expression content, toxicity, incapable of fresh edible and long growth periods. तो आपको पता है जो एल्गी है उसका ग्रोथ पीरियड लेस होता है. तो एक तो ये है कि हम इसको we can produce the edible vaccine in very less time. तो आपका ये एक तो टाइम को रिड्यूस कर दिया है और फ्रेश we also can use. Okay, then at long last algae can effectively lyophilize and when dried can be stored at room temperature for 20 months without losing antigenic efficiency. इसके अलावा क्या है, other than this these algae if we dried and prepare a dried powder then it can be stored for 20 months. Month means it show the durability. So the algae well system is solve the keeping quality, large amount, So this system is a now ideal and best system for developing the edible vaccines. Also, there are some systems like insect-based edible vaccines. So the VVS Vaculovirus Expression Vector System and insect cell culture innovations were acknowledged and also used as a alternative option for generation of recombinant protein. तो अभी रिसेंट में BEVS Baculovirus Expression Vector System and your आपका insect cell culture is also used for the development of edible vaccines. Also, the whole cell yeast-based vaccine, जो आप already last slide we had discussed, that 2 clinical trial being developed. GS this 4774 for this hepatitis B and GI 5500 for hepatitis C. These clinical already going on and these are in phase 2nd. So this is a good system for developing the vaccines, and lactic acid bacteria based vaccine is also developed. This delivery system can provide protection against deterioration of the antigen and trigger both innate and adaptive immune response. Means जो lactic acid bacteria है, means in the milk, via milk we are giving the edible vaccines, so it will trigger both innate का मीन्स जो आपको वर्थ से आपको जो इम्यूनिटी मिलती है दैट इज इनेट, एडेप्टिव जो आप सम एक्वायर्ड करते हैं, एक्वायर्ड विद द एनवायरमेंटल एंड कंडीशंस नोन एज एक्वायर्ड इम्यूनिटी, तो दोनों इम्यूनिटी के अगेंस्ट वर्क करता है. सो दीज आर द सम सिस्टम्स लाइक there are so many systems which are used for the developing edible vaccines, and first one is the vegetables like tomato, potato and carrot cells which are used for the developing edible vaccines. Then fruits, your banana is the best example where we are using the developing for developing edible vaccines. Then algal based system, insect based system and the lactic acid bacteria system. So there are more system, but the yeast system is the very good system because it producing the large quantity and the keeping quality is also high. So this is the best system for developing the edible vaccines. Now the production and purification. So what are the important points which are considered for the vaccine production and the purification of these edible vaccines? So transgenic plant vaccine candidates. So use of plant to produce plentiful foreign antigen proteins and then manufacturing into vaccine after purification and separation. So this is the first process कि जो आपका foreign antigen protein को जब हमने ट्रांसफर किया है तो आपका जो प्यूरिफाई होना चाहिए and after separation because there will be so many secondary metabolites will be also there, and use of fresh edible plants or plant tissue to convert the interest of proteins. सेकेंड ये है कि हमारा जो वैक्सीन है एडिबल वैक्सीन वो मेनली फ्रेश और एडिबल प्लांट पार्ट होना चाहिए दैट विल बी हेल्थी एंड न्यूट्रिशियस. थर्ड वन इज द प्यूरिफिकेशन एंड दिस इज द मेन रीजन बिहाइंड द हाई कॉस्ट बिकॉज इट टेक्स सम कॉस्ट एंड देयर आर मेनी प्रॉब्लम फॉर द प्यूरिफिकेशन सच एज इन्फ्लुएंस ऑफ पॉलीफिनोल्स, alkaloids and other secondary metabolites. So these secondary metabolites affect the your final edible vaccine quality. So it will be need and it have higher cost. And use of protein based polymer with inverse temperature transition to purely insulin expressed by the chloroplast vector. So this is the method, means we use some centrifugation and vectors and inverse temperature, and also we use some polymers to purify the proteins or your vaccines. And foreign protein expressed in seed assure excellent storage properties. आपका जो सीड है जैसे क्रॉप्स है वो आपका excellent storage property होता है, and thus added flexibility in processing management and both productions. और वो आपका जो प्रोसेसिंग है और प्रोडक्शन है दैट इज आल्सो वेरी इजी बिकॉज एक्सट्रैक्शन इज वेरी इजी फ्रॉम द सीड्स. यूज ऑफ or polymer fusion to facilitate purification of recombinant protein. The oleosins and other polymers are used for the purification of recombinant protein because they bind and polymerize the proteins. Assembled protein of edible tissue reduce the cost. Oral delivery. Example, oral delivery. Generally, expression content of most recombinant protein in plant is 0.01 to 0.4 of total soluble protein. So there are certain points like what are the points which are considered before the production and purification of edible vaccines, like they have seed assured proteins are good. Also oleoresin or polyresin used to for the fusion of the proteins, and also we need the phase and the quantity is very less. Now there are some biosafety or regulatory issues which are also considered before the use of this edible vaccines. So first is the inadequate information, means we need full information before the use of this transgenic vaccine, that what are their current impact or what are their future impact, and about the plant capacity to post transcriptionally modified human proteins, means after the taking, what changes they can do in your human body, means there are some post transcriptional or translational changes which also can be modified the human protein. So we need full of information before the use of this edible or transgenic vaccines. And induced tolerance as repeated exposure. If we will repeatedly use this edible vaccine, then there will be a chances to induce the tolerance for the particular virus. Means in the after some, may be possible that after some time the high dose of this vaccine also will be ineffective against these viruses. So it will also regulatory issue before the using these vaccines, and should administered appropriately, means there will be a system to development and deliver of these vaccines, means they will be administered properly. Solutions. In many cases, they induce the male sterility to overcome issue of pollen outcross and biodiversity. So in the some case there will be a scene of male sterility, so they affect your pollen or fertilization and biodiversity, so many factors they affect. Use of tissue specific or inducible promoter to prevent adverse effect on the growth, development or environment, means we have to use only specific or inducible promoter, so the other adverse effect will be minimized. And utilization of chloroplast by maternally inherited to maintain pollen mediated gene flow, means if we will use the chloroplast or maternally inherited your antigens, then there will be less male sterility. Designing plant good manufacturing factories and waste product disposal. Also there will be a need of developing some good manufacturing factory or designing a balance or designed manufacturing factory and waste product. You know that this is bio waste, and there will be a rules or regulation for the disposal of this bio waste. So there are some biosafety and regulatory issues, means आपका पूरा इंफॉर्मेशन होना चाहिए, और अगर ऐसा नहीं है, कहीं ऐसा तो नहीं है कि बार बार लेने से उसके लिए टॉलरेंस आ गया हो, तो दैट इज आल्सो ए कंसर्न, एंड द all the process should be administered properly, means there will be a system to regulate, and there will be a male sterility course, so maybe possible we can use the maternally inherited जो आपका एंटीजन स्पेसिफिक है वो यूज कर सकते हैं. ऑल्सो एक स्पेसिफिक प्रोमोटर यूज करो जिससे कि वो अदर चीज पे भी इफेक्ट ना हो, ना कोई उसे इफेक्ट करे. ऑल्सो देयर विल बी ए गुड मैन्युफैक्चरिंग फैक्ट्रीज एंड ऑल्सो वेस्ट प्रोडक्ट डिस्पोजल. So these are the some revenue. Also there are some examples like antigen antibody system is the known system which induce the immune system. Okay, So HR, there will be a antigen for this rotavirus transferred into the potato, that is known as HRVBP7. IgG, immunoglobulin G for hepatitis B transferred in the tobacco. LTB for the in the maize and potato for the diarrhea. Tetanus C for tetanus vaccine transfer in the tobacco, chloroplast. Immunoglobulin G again for rabies virus transferred into the tobacco. CP protein and potato for Norwalk virus for diarrhea. Rabies virus in spinach. HIV1 for the TMB virus and through the spinach, means we use TMB as a virus system to developing this edible vaccine. So there are some example, and these trials are in the some phase 1 and phase 2 phase second. So trial is going on, and may be possible that in the future we will get the edible or transgenic vaccines which are easy to take and control some viral diseases. Means if you will eat one potato, there will be no rotavirus, or it will provide immunity against the rotavirus. So these all example, and there are trial in some advanced phase. So this is about the transgenic plant vaccine, that what is the transgenic vaccine, and what systems used for the developing this system, and what are their advantages, their disadvantages, and what are the regulatory issues which will be considered, and there are some example. So this topic is very important for the ARS and CSR exams, and the both in the mains exams also they can ask some question, because edible vaccines are the major concern, because now only some last year we faces some coronavirus, and there are so many vaccines, and the vaccine is the very burning topic for the competitive exams. So hope you understand, and thank you very much.